about urea cycle. Urea cycle is also called the ornithine cycle. The urea cycle is a liver process that converts toxic ammonia from protein breakdown into urea, which is exerted in urine to remove excess nitrogen. The urea cycle is a metabolic process that takes place in the liver. Its main function is to remove excess nitrogens from the body. When we eat protein, it breaks down into amino acid. During amino acid breakdown, ammonia is released, which is toxic to the body. To prevent ammonia buildup, the liver converts it into urea, which is eliminated through the urine by the kidneys. So let's give an example of urea cycle. So just imagine you just eat chicken, which is the chicken is rich in protein. So the number one is protein breakdown. So your body digests chicken into amino acid. The second one is amino acid metabolism. So during use of amino acids for energy, a toxic byproduct called ammonia is released. So the third one is ammonia detoxifications in the liver. So the liver runs the urea cycle to safely remove this ammonia. And the carbonyl phosphate combines with ornithines, it forms centrulline. And centrulline exists in the mitochondria and combines with aspartate. It forms argininosuccinate. Then argininosuccinate splits into arginine and fumarate. Arginine breaks down, produces urea and regenerates ornithine, which re-enters the cycle. Urea goes into the blood and filtered by the kidneys and exerted as urine. So let's proceed to the ester bond. Ester bond. An ester bond is a type of covalent bond formed between a carboxylic acid and an alcohol. It occurs when the hydroxyl group of the carboxylic acid react with the hydrogen atom from the alcohol. These reactions result in the release of a water molecule and the creation of a new linkage called an ester bond. This reaction is known as condensation or dehydration synthesis reactions because water is removed during bond formation. So, this is the general formula of ester bond. Okay, so let's talk about how ester bonds work. So, the first one is formations, which is condensation. Ester bonds are created by the removal of water between two molecules. For example, in triglycerides or fats, 3 fatty acids join with one glycerol molecule through 3 ester bonds. Second one is breaking, which is the hydrolysis. Ester bonds can be broken by adding water back into the molecule. This process is called hydrolysis and is usually assisted by enzymes such as lipase. During digestion, hydrolysis reverses the reactions separating fatty acid from glycerol. Ester bonds have multiple biological and chemical functions. So, let's talk about functions of ester bonds. So, the first one is the energy storage. In living organisms, ester bonds hold fatty acids to glycerol, forming fats and oil. These fats serve as major energy reserve because ester bonds store a large amount of chemical energy that can be released when broken down. The second one is a structural rule in cell. In phospholipids, ester bonds link a fatty acid to glycerol. Phospholipids are essentials for forming cell membranes, which protect the cell and control what enters and exits. The third one is the biological importance. Ester bonds allow the formations of large, stable molecules such as triglycerides and phospholipids. Without ester bonds, cells would not have strong membrane and organisms would lose a vital way to store energy. So the fourth one is the industrial and everyday applications. Esters are made from ester bonds, often have pleasant smells and are widely used in perfumes, flavorings, and food additives. They are also used in solvents, plastics, and pharmaceuticals. I think that's all, and thank you for watching. Hope you enjoy my video.